Hey all, welcome to ShareTrek. This is Raj here. I'm carrying forward the discussion I had in the last video with you about genetic variations and how the next frontier would be to narrow them down and take appropriate editing technology and delivery technology to tackle them, and how it would lead to higher probability of success and lower cost for therapies. In this video, I'm going to share exact details of what has been accomplished using artificial intelligence and other methods, and what is coming next in this quest to narrow down the potential candidates of genetic variations that are linked to various human disorders. So with that said, let's get started. Welcome back, friends. Kyle Farr, clinical geneticist at the DNA sequencing company Illumina, has been a key player in a landmark research of gigantic proportions and complexity. Given that human genome sequencing has turned up myriad variants of individual genes that might explain disease or treatments, but human genetics alone often can't tell us whether a variant is medically relevant, Kyle has helped carry forward the investigation into narrowing the long list of variants so that clinical targets can be picked up more effectively. In 2019, Kyle reached out to evolutionary geneticist Thomas Marques Bonet from the Institute of Evolutionary Biology in Barcelona, Spain, and he also reached out to primate geneticist Jeffrey Rogers at Baylor College of Medicine with a proposal. He said that if they could come up with blood samples from multiple members of many of the world's 500 plus primates, Illumina will help fund the DNA sequencing. This was the first daunting step because, first of all, many of these primate species are endangered and protected. Next, there is a lot of paperwork and procedures to get permission from various governments and authorities to collect the sample, and finally the primate has to be sedated to collect the sample. Just to give a perspective, 60% of non-human primates are classified as endangered species and come under special protection, and therefore there is a lot of red tape that has to be crossed in order to get the samples. Marques Bonet took up the task of signing up researchers worldwide to commence the task of doing the paperwork and collecting samples from primates in their countries. One of the biggest contributors to the research was Jean Bobley, who contributed samples from 77 South American species by collaborating with local scientists, museums, and zoos in South America. Meanwhile, in India, Govindaswamy Umapati, who is a conservation biologist at the Center of Cellular and Molecular Biology, traveled from state to state in India to lobby forest managers and local officials for access to gibbons, lorises, macaques, and lemurs in order to collect samples. One of Marques Bonet's postdocs, Lucas Kuderna, who now works at Illumina, led the consortium and sequenced 703 individuals of 211 different species using short-read technology. The new data joined 106 already sequenced genomes from 29 additional primate species. Added to this was a set of genomes from 27 other primate species provided by a geneticist at the Chinese Academy of Sciences Institute of Zoology. The availability of primate genomes provided Farr, Rogers, Marques Bonet, and their team with an opportunity to search for single nucleotide polymorphisms, or SNPs, which are DNA-based variations occurring within or across a species. These SNPs have the potential to modify the proteins produced by the genes or impact the functioning of a gene. The researchers discovered 4.3 million SNPs that cause changes in the amino acid sequence of proteins. Single nucleotide polymorphisms, or SNPs, are tiny variations that occur in our DNA. To understand SNPs, let us start with DNA, which is the genetic material that carries instructions for building and functioning of our bodies. DNA is made up of a long sequence of building blocks called nucleotides. Now imagine that these nucleotides are like letters in a generic genetic alphabet, and a single nucleotide is like a single letter. SNPs happen when there is a change in just one of these letters within our DNA. For example, instead of having the letter A at a specific position, SNP might result in the position having the letter C or G instead. These tiny changes in our DNA can occur naturally and can be found in different individuals. They are what make us unique and contribute to our individual differences like eye color, height, or susceptibility to certain diseases and potentially allergies. SNPs can sometimes have effect on our bodies that can influence how our genes work, which are the instructions for making proteins. Proteins are the molecules that perform many important functions in our body. So if a SNP occurs in a gene, it might change the protein that is produced or affect how it functions. Scientists study SNPs to understand how they can impact our health, how we respond to medications, and even our risk of developing certain diseases. By exploring these tiny variations, the researchers can gain insights into genetic factors that contribute to our unique characteristics and overall well-being. Assuming that variations in our DNA called SNPs are commonly seen in both humans and other primates without causing diseases, Farr and his team cleared many human variations from suspicion. They developed a computer program called Primate AI 3D, which uses a tool called AlphaFold based on artificial intelligence to create 3D models of proteins. By analyzing the harmless SNPs found in primates, the program identifies parts of the protein structure where changes would not disrupt its function. On the other hand, changes in other regions are more likely to cause problems. Next, they use the program to predict the potential harm caused by human SNPs. They compared these predictions with a database of human DNA changes that were tentatively linked to diseases. As a result, they concluded that around 6% of the SNPs that were tentatively linked to disease were probably harmless. The team also used primate-trained AI to do the opposite, identify harmful genes. They applied it to the health records and gene variant data of 454,712 people in the UK Biobank in order to find SNPs likely to play a role in 90 different human health concerns. Now, Primate AI 3D, a deep learning network trained on 4.5 million common genetic variants from 233 primate species, is available. Uncovering the role of rare but pertinent variants in common diseases is crucial for personalized medicine and can facilitate drug development. Primate AI 3D addresses the earlier inability to predict variant pathogenicity, which has hindered efforts to study these rare variants, and additionally, it now enables identifying pathogenic genetic variants underpinning diseases and can reveal novel drug target candidate genes, further increasing the clinical actionability for such variants. And that's the benefit of having Primate AI 3D with all those AI trained data sets. This is deep value in Illumina, and in my opinion, this sets it apart from its competitors. The offshoot from this AI model will provide further growth catalysts and opportunities for Illumina down the road. And as the AI gets trained on more and more data, it is going to get even more powerful, and it will be giving even more powerful inferences and data that can form catalysts for Illumina. And also, it would be helpful to various genomic companies in identifying drug targets that are more reliable indicators for solving a particular disease condition. And it will also reduce the possibility that a particular therapy might be found ineffective in phase 2 of the clinical trial, thereby saving a lot of money for the company and also saving a lot of time for patients, because genomic companies will be able to avoid going along the wrong path for a long period of time and spending lots of dollars and adding to the cost and the duration taken to deliver effective therapies. So these are all going to be the advantages of having Primate 3D AI kind of systems looking at all the variants that are happening in human DNA and trying to narrow down the list of variants that have to be looked at in order to treat various genetic conditions. And even in that, if you are looking at SNPs, then they are more amenable to something like prime and base editing instead of doing CRISPR-Cas9. So basically, I think that the availability of AI systems like Primate 3D AI and the fact that Illumina is going to provide those software to its clients is going to help genomic companies not only to reliably identify target candidates for gene editing, but also it will give them certainty that if those sections are edited, it is going to solve a particular problem. And it will also give them a clear idea of which tool they need to use in order to address those genetic editing, and also it will help them to identify more reliably which transport mechanism to use to deploy those therapies into the body. So I think that's what is the utility of this particular exercise for Illumina. It's going to be a force multiplier for it. From an investment perspective, that's what I look at and conclude. And meanwhile, I'd also like to know what you guys think about Illumina and this particular investment that they have done in propagating primate 3D AI. So I would like to look forward to your comments. I'll look at the comment section to get some insight about what our audience thinks. I overall feel that Illumina is, despite all the boardroom problems and the Grail struggle, it's still at the forefront of genomic sequencing, NSG. And I think with that, I would like to end this video. That's all from me for now.